our department there are two types of electron microscope are available. One is transmission electron microscope and other is scanning. In transmission electron microscope, electron transmit through the specimen, but in scanning one, electron does not transmit through the specimen, it just scans the surface. This is scanning electron microscope laboratory. Here I will discuss about the scanning electron microscope. This is Quanta FEG 450. This is scanning electron microscope. Model is Quanta FEG 450. Here, electron emits from the source, passes through the various electromagnetic lenses, then it falls on the specimen where there is an emission of secondary electron. These are the specimen on the aluminum stem, where whatever the material, whether it's a biological or material sample, we place our material on the aluminum stem with the help of conducting carbon tape. Then we place here in the specimen chamber, where whenever the electron hits on the specimen, there is an emission of various signals. Secondary electrons emission are there, back scattered electrons are there, and there the X-ray signals are also emitted there. But for secondary electron emission, we record the images which are analyzed in the machine. This is the platinum nanoparticle. Here we can see shape as well as the size. Shape is the spherical one and size is in the range of nanometer. Here you see 50 nanometer, 50 nanometer. So by this machine we can find out the shape, size and surface is spherical. And other thing, we can also find out whether it's a platinum material or some other element. For that we perform the EDAX analysis. Here you see, this is the EDAX spectrum which are recorded during analysis. You see platinum elements are there. This confirms whatever the nanoparticles synthesized by our scientists, we can confirm by the EDAX analysis. Okay? These are some other samples. This is the tetrahymena sample. We can see the surface morphology here. This is the mouth organ, locomotion organ is there, and this is the fungal sample are there. You see the fungal spores are here. So variety of material can be analyzed by the scanning electron microscope. So this machine is used for shape, size, surface morphology, as well as what are the elements present in the specimen that can also be analyzed very easily.
This is our 120 kilovolt transmission electron microscope. It is the make of FEI company. This is our sample injector. Here we load the sample. Here, this is the grid. This is the grid. This is basically the slide for the transmission electron microscope. Its name is grid. This is made up of carbon, and it contains 200 holes. This grid is 200 mesh grid. The samples is loaded here, and this grid fits into the clamp of the sample injector, and this sample injector goes inside the goniometer and it fits into the column of the machine. In the top of the column there is a electron source called lab 6. The electron beam emits from the electron source. It passes through different electromagnetic lenses and it falls into the samples, and the electron beam transmits through the samples, and in the fluorescent screen the image is appeared. The basic principle of this microscope is the electron beam must be transmitted through the samples, then only the image will be formed. Final image formed in the fluorescent screen, and from here we transfer the samples, the image into the computer, and from the computer we can enhance the magnification up to the 8 lakh times, and we can achieve the resolution up to 0.1 nanometer. These are the different types of samples. You can see this is the cell line. We can see the ultra structure, the internal structure of the cell lines. This is the Leishmania samples. This is the sample of the chloroplast. This is the brain myelin sample. This is the kidney glomerulus samples. Means you can see the internal structure of a cell or tissue through these samples, through this microscope. You can also analyze the nanoparticle or the materia samples. You can find out the shape size and the morphology of the sample through transmission electron microscope also.
This is confocal microscope. This is the SEM-880 model based on the fluorescence optics. This instrument contains some control panels for switch on the instrument. This is control panel for the confocal microscope. This is control panel for the fluorescent lamp, and this is for the laser sources. And these are the binocular lenses for this microscope. This is kind of the inverted microscope, so we fix the slide in upside down position in this microscope. This is a stage where we put our slides. This is our slide, and we put the slide in upside down position. And this is a joystick. By using this joystick, we move our slide in the XY position. By using this control panel, we generally select the objective lenses. This contains the 10x, 20x, 40x oil immersion, 40x water immersion, and 63x and 100x. In this slide, we use the 20x location, and using this locate switches, generally we locate our slide, and by setting on the shutter, these are some channels which are the refractor lenses. First and last positions are empty, and the second is filled with the GFP, third one is filled with the DS-RAD, fourth one is filled with the CFP, and fifth one is filled with the DAPI. I choose the DAPI position and locate the sample. I locate the sample by using this joystick to move our sample in the XY position, and using this coarse and fine focusing. I just focus the sample, and then off this shutter, then go to the acquisition position. Then I on the laser, and make the channels. In this slide, we use the Alexa 594 and Alexa 488 and the DAPI channel. and use the bad signal because there is no bleed through. In the fastest mode, there is the chances of bleed through. In the smartest mode, we got the bright images. So we choose the best signals and apply these. This is the how we capture the image of by using this confocal microscope. In this microscope, we use the laser sources as the illumination source. When the laser light hits the sample, it excites the molecules and it emits some light, and which light is by going through the dichroic mirror detected by the detector and image is generated. This is the basic principle how we generate the image from confocal microscope. 7 lines in this confocal microscope, which are 458, 488, 514, 405, 561, 594 and 633. These are the main laser lines which we are used. And in this instrument we also have an attachment which is known as the Airy scan and also called as the super resolution. By using this Airy scan or super resolution, we got the resolution up to the range of 140 nanometer. This confocal microscope captures the more specific and precise reason images, and because it uses laser sources as an illumination source, and it contains the 2 pinhole apertures, and first aperture is after the laser lines, which specifically illuminates the specific region of the sample, and second pinhole filter is located before the detector, which cut the out of focus light, so we got the specific region images very appropriately. The application of this confocal microscope is used to image or localize the protein or macromolecules which are fluorescently tagged by the fluorescent dyes or antibodies tagged with the fluorescent dyes, and it also used for the live cell imaging, and we use this instrument for the reconstruction of the 3D images. Some images which are captured by this microscopes are, this is live cell imaging, some Drosophila, and this is also the live cell imaging. These are some pictures. This is 3D image. This is also the image generated by this microscope. This is also the cytoskeleton tagged by the phenolene dye. These are the pictures. This is the metaphase stage. These are some pictures. Thank you.